Thanks, Dennis. Dennis asked us for these titles last November. He hits us up. He's like, Peter, it's your turn to do a talk again. I'm like, okay. What are you going to talk about? I'm like, well, I'll talk about bacteria and the river and something to do with indicators because that's what we're working on lately. So I came up with this title, Bacteria in the Indian River Lagoon, Indicators of Health. And it was really when I started to put this talk together that two things occurred to me. Firstly, I now have a graduate student who can do half the talk for me, which is a really good thing, because then I only have to put together a 25-minute talk. And the other thing is we're talking about health. And what do we mean by when we're talking about health? A lot of the talks in this series have talked about our impacts on the health of the environment. So how are we impacting the health of the Indian River Lagoon? But part of our work is also looking at the lagoon and how it can impact our health. So looking at the presence of pathogenic bacteria in the river and what hazard they may cause for us, okay? So we've got two parts to this talk. I'm going to start off going all the way back to the beginning, talking about bacteria, what they are, how diverse they are, talking about bacteria and humans, then getting into what are indicator species and how we can look for these indicator species within the Indian River Lagoon and what that may imply in regards to the health of the lagoon. Then I'll pass over the whole talk to Gabby, and then she will be talking about pathogenic bacteria in the IRL, and then we'll just wrap up and you'll all be free to go out for snacks or whatever it is you do after this. So, what is a bacterium? This is not a bacterium. This is a mammalian cell. It's a typical human type cell. It has a nucleus, it has mitochondria, it has a membrane around the outside. So it's a fairly complex cell. This is what you are composed of. By contrast, this is a bacterium. Okay, you see that little thing over there? And I did this to size scale. So a typical mammalian cell is anywhere from 10 to 30 microns across. A bacterial cell is somewhere between 1 and 2 microns. So they are far, far smaller. Of course, we can't see that, so we use a microscope and we make them bigger. So this is a typical bacterial cell. It's a small, simple cell. It has no nucleus. Nucleus there, no nucleus there. It has DNA, obviously. It has a cell wall. It has some other little structures inside the cytoplasm, and it may well have something like a flagella or a group of flagella which allow it to move. Okay, so they're simple cells. So you look at that and go, oh, it's a bacterium. All these bacteria must be the same. But as soon as we start looking at a lot of different bacteria down the microscope, the first thing we see is a whole bunch of different shapes. Okay? So at the top here, we have what's called a coccus. It's a little sphere, simple little bacterium. And this is an example of a coccoid bacterium called Enterococcus faecalis. MRSA is also a coccoid bacterium, Staphylococcus aureus. Okay? We can also get rods. So this is a rod. It's a thing called a bacillus. And something like Escherichia coli is a rod. These are all scanning electron micrographs. They're all false colored. EMs don't come out color like this. People just take time to make them pretty. Because bacteria are pretty, but they're not colored like this. So this is E. coli. You'll have heard of E. coli, I'm sure. We also see things which are curved rods. So if you take a rod like this and you curve it slightly, then you have something which is called a Vibrio. Okay, a different type of bacterium. Some of these things are very complex. So we see spirochetes. This is a spirochete, and this is what it looks like under electron micrograph. And you can see it's like a little corkscrew. And this is something we see this type of bacterium in the river, but this particular bacterium is Borrelia, and this is the causative agent of Lyme disease. Okay, really pretty little bacterium, but then I'm a microbiologist. So we see diversity at the level of just looking at them for their shapes. But if we want to really find out about diversity, we can go to a molecular level. We can start looking at what is their genetic makeup. And that's what this diagram is. This is the tree of life, and it's described by a guy called Norm Pace a few years ago now. But it's based on the diversity of a single gene type, which is in all these forms of life. To orient you, you are here. Okay? So this is man, right there, at the end of the tree. Right next to you is Coprinus. Coprinus is a mushroom. On the other side of you is Zea. Zea is the maize plant. So most of the big diversity that you see in this planet, when you look out the window and you go, wow, this is such a diverse area we live in, all the trees and birds and fish and everything else that's running around, it's this tiny little bit at the end of the diversity tree. Everything else that we have here, these are eukaryotes, these all have nuclei, but it's things like slime molds and protozoa. They're all there, they're all microscopic. Then at the top of the tree here, these are the prokaryotes. These things do not have nuclei. They're the simple little cells. And this is divided into two groups. We have the archaea and the bacteria. The archaea are simple microorganisms, highly diverse. They are fascinating. The first places we found these things was in very high temperature environments. We're now finding them throughout the oceans. They're very common. Very diverse group. One day I'll give a talk about them. But we're talking today about the bacteria. And you can see just from this tree how diverse bacteria are. Bacteria are very ancient. They were the first form of life on this planet. So they've had something like 3.8 billion years to evolve. And they have evolved to inhabit all the niches on this planet. Okay? I've spoken before about the oceans. I do a lot of work with marine sponges and the bacteria and other microbes that live inside the sponges. If you were here in November, you would have heard a fascinating talk by Jennifer Biddle, who was talking about the bacteria that live in the deep subsurface. So you can drill down into rock. You can go down, way, way down in the rock, and you will find bacteria living down there and thriving in those environments. Bacteria are also highly adapted to growing in association with plants and animals. Of course, we're just another animal. You might well have heard about the Human Microbiome Project. This is a project which is designed to work out how many different types of microorganisms are associated with our bodies. So they are taking samples of various locations throughout the body and then using molecular biology techniques to work out what is the diversity of microbes associated with that site. This is the version of the chart which is from Science. This is the version of the chart which shows up on the Human Microbiome Project website. But we can see from this that we have about 600 or more species of microorganisms which are associated with your mouth into the lungs. Large numbers of bacteria. On the skin, there's 1,000 different species. When you look in the stomach, which is not a very hospitable environment, it's very acid, you've still got 25 different things living there. And then if you get into the intestines, there's another 500 to 1,000 species there. So you can always remember, no matter how lonely you're feeling, you have a bunch of friends with you. The bottom line on the Human Microbiome Project is that there are at least 10 times as many bacterial cells in the body as there are human cells. So all your cells are outnumbered 10 to 1, okay? Bacterial cells account for 1 to 3% of your total body mass. They're very small. So even though you've got 10 times more than you do human cells, they're still not, they're still about 1 to 3% of the total body mass. On a good day, I weigh about 180. So I'm carrying around 5 pounds of bacteria with me on a daily basis. Something to think about. Not long, but think about it. The human microbiome is really important, and this is what people are really finding out now. It has a huge impact on your health, okay? They're now finding that you have to have this intact microbiology associated with your body because it is involved in processes like weight gain. If it goes out of whack, you're going to gain weight. It's involved in the aging process. It's involved in cancer progression. It's certainly involved in infectious disease. So when you're put on antibiotics, you really mess up your normal microbiome, and that can allow other microbes to start to grow. But for the purposes of today's talk, we're really focusing in on the intestinal flora. This is all the microbes that live inside your digestive tract. It's found in you, it's found in all animals. And it carries out many, many different functions, including the digestion of carbohydrates that you cannot digest. They're really helping you in that process. You eat something, you can't digest it, the bugs can. They make little fatty acids, which you can then absorb. So you're working in symbiosis with these microbes living inside your gut. And there they are certainly involved in the repression of other pathogens. There are about 500 to 1,000 species of bacteria living inside your intestines. And those include Escherichia coli and Enterococcus faecalis. These are not pathogens. These are things which are living there symbiotically and doing something really good for you. Okay? When we are talking about indicators of water quality, we're talking about the process which is used to work out how safe drinking water is, recreational water is, what's happening with wastewater, how efficiently is it being cleaned up. And to be able to study this over the last 100 years, people have developed tests to look for fecal indicators, which are a group of organisms which indicate the presence of fecal contamination. And this includes the coliforms like E. coli and thermotolerant coliforms, and also enterococci. So these are things which are commonly present in your intestines. They enter the fecal matter. They could get into the water supply, but they're an indicator that the fecal material is there. There are many standard bacteriological methods which allow us to test for these, and these are universally used. So I can test water here, and it's comparable to data that's generated in Europe or Asia or anywhere. So we can get similar numbers which all are comparable. The result of these sort of tests are these warning signs. So it's like, this was from down in Martin County over the summer. It's like, don't go in the water. We've got high numbers of bacteria. The bacteria they're referring to are these fecal coliforms and enterococci. Beaches may be closed. Water unsafe for bathing, which these people are promptly ignoring. But the state has strict guidelines on how we can interpret these tests. So for fecal coliforms, they have distinct numbers which say the water is good if there's less than 200 CFUs per 100 mils, and it's poor if there's over 400. For the enterococci, the numbers are lower, so 35 or less is good, over 105 is poor. So we have these guidelines, we know where we're working towards. If we're trying to test anything for a bacterium, we go through a simple process. We have a sample, we then inoculate it onto a growth medium. The growth medium can be very selective, so it's only growing certain things. We then incubate it, and then that gives us growth on a petri dish. Oh, okay, so we get water samples from the river. So we're interested in what sort of levels of bacteria are present in water samples. The water samples come back to the lab. This is Kathleen Janda working away in my lab. The water samples come in these bottles. The first thing we have to do is some dilution series, because any bacterial sample, any water sample will contain anywhere between 100,000 and 10 million bacteria per milliliter. So Cath does a dilution series, and then they can be filtered. Even though these things are really small, we have filter systems which are able to trap these small bacteria on the surface of the filter. And you can see here the water is being pulled through under vacuum. This is the water, which is waste. The filter is on the top here. The water is pulled through. And what you'll see now is a filter which has bacteria stuck on the surface. That filter can then be put onto a petri dish, which contains a specific growth medium, and then that is just popped into the oven, and 24 hours later, we get some data. So we are using 2 different growth media to be able to look for these coliforms and enterococci. We use MEI medium. Here you can see the filter after it's grown up, and you can see the little blue dots. Those are colonies of enterococci, which we can count. So the blue colonies are enterococci. On MI, we can count the total number of Escherichia coli colonies, which are blue, and under UV light, we can see fluorescent colonies, which are the other coliforms. So by combining these numbers, we can get a measure of total coliforms, and by counting this, we're getting a measure of the enterococci present in a water sample. The sampling sites we're using are the ones which Dennis Hanasak has been using as a long-term study sites. So they range from Vero Beach all the way down towards the Fort Pierce Inlet, going from North Grand Harbor, Vero Isles, South Canal, Oslo Road, Linkport, which is where we are now, down to Torpy Road. And we can look at these sites in terms of ones which are highly impacted, because these are within the city of Vero Beach, and they have influx from the North Canal, the Main Canal, and the South Canal. They're also distant from the inlet, so they're not getting a great deal of exchange. The southern 3 sites are comparatively healthy, theoretically, and so here there is no major input, and they're closer to the inlet, so they're getting exchange going on. So let's look at some data. This is the top study site, northernmost, North Grand Harbor, and this is looking at total coliforms. So this is the total number of coliforms in CFU's colony-forming units per 100 mils over a period from October of 2011 to whatever that says. I can't read that. January 30th, thank you, you can read that. You can help me out. But this is over the course of just over a year. And you can see immediately, you look at this and you go, wow, there's a low period here, and there's a high period here. And this corresponds to tourist season and hurricane season, the 2 seasons we have in Florida. Thankfully, we can also get rainfall data. Okay, this is from the St. John's Water Management District, and we can get rainfall information. So this is a miracle because this actually correlates beautifully. So we get high rainfall here, high coliforms, low rainfall in tourist season, and then we go into the wet season and we see an increase in the amount of coliforms in the water. So there's obviously some impact going on from rainfall and a correlation with the amount of bacteria we see in the river. We can take that out to a longer time period. We've been doing this work since October. This was our first collection time, and this goes through the December of last year. This is from the northern sites, so this is just taking an average of the readings for each of the 3 northern sites. And you can see dry season, wet season, and then this is a year later. This is the data for the southern sites. And again, you can see dry season, wet season, and so on, okay? Interestingly, this was the dry season I was just talking about, and it became obvious that the fecal coliforms were very low at this time. If we look a year later, this is an equivalent time when I was hoping to see this seasonality repeat. It didn't. And the reason for that was we didn't have a good dry season last year. It was a wet, dry season. So there was rainfall, and we didn't see this drop back down. So we're continuing with this monitoring. We're hoping that eventually we'll get back to see what the true seasonality is of this data. I showed you this earlier. This is the state interpretation of this data. And if we put along a line, which is poor water quality, it comes there. Okay? So during the one dry spell we had water qualities according to fecal coliforms which would be considered good to moderate. The rest of the time, according to coliforms, the water quality is generally very poor at all these sites. We've done a similar thing with the enterococci, okay, but we get a different data set. It's very interesting. Same time period, October 11 to December 13. Here's the dry period. Not the same data. We see different things going on. This is the following dry season, different set of data. And this line is the cutoff for poor water quality according to enterococci. So from doing two different tests, we get two totally different results. So fecal coliforms, it says the water quality is generally very poor, except during dry spells. And for the enterococci, the water quality is generally good, except for short periods with high counts. So what does that mean? If anyone knows, let me know. When we're interpreting this data, we have to keep in mind one very, very important fact. If anyone wants to buy a copy of this book for the library, I would be very happy. It's available on Amazon, and in this, Taro Gomi describes for 3-year-olds that everybody poops. Okay, so humans poop, horses poop, cows poop, pigs poop, zebras poop, ducks poop. It's a whole book full of poop. But in scientific terms, this is significant. Okay, because we can talk about coliforms and enterococci, they are seen in association with the intestines of animals. We are animals, but so are ducks, cows, dogs, cats, and humans. This is really quite interesting. We're down the bottom here. This is not in any particular order, but down the bottom you can see that per gram of fecal material we have something like 13 million coliforms and 3 million enterococci. Cows, which you might think of as being rather dirty and disgusting, have less. Okay? Dogs, look at that. Nearly 1 billion enterococci per gram of fecal material. So if any of you have dogs, please clean up after your dogs. Okay? But we really, when we're interpreting this water data, we really don't know what the source is of these fecal coliforms and enterococci. We want to try to find out something like this. And this is where we're heading to right now. We're going to be doing this over the next few months. But if we're thinking about the river, what are the sources of fecal coliforms and enterococci? Yes, they could be coming from septic tanks and sewage treatment plants. They could be human waste. But they could also be coming from agricultural runoff. We have a lot of farmlands just inland. Those farmlands, water washes down the canals straight into the river. So that's a potential source. We also have a lot of wildlife. We're very fortunate to have a lot of wildlife here. A lot of birds. Birds all have these bacteria associated with them as well, as do aquatic mammals. No one has determined how many enterococci or fecal coliforms a manatee has. But there's a lot of manatees out there. So we need to be able to work out where the material is coming from. And this is a process called source tracking. And these are molecular techniques which allow us to track specific microbes and indicate their source. And the current recommended method for human bacteria, human waste, is to use a molecular technique which determines the amount of Bacteroides HF183 in a water sample. And so this is what we're going to be setting up over the next 6 months to try to start to dissect out some of this data. Further down the line, we'll also need to set up things like cow and bird. There's a whole series of these tests which allow people to see where contamination in water bodies is coming from and what it might actually mean. Another side of the work we're doing with the bacteria is just looking at the total number of bacteria which is present in a water sample. Okay, so this is what is called total heterotrophs. This is not a terribly selective method. We're just trying to get an idea as to what the bacterial load is in these water samples. So to do this, we take a water sample, we dilute it, we plate it onto a specific medium, incubate it, and then see what grows. And after a few days, you get these multiple colonies, you get lots of different types of things growing, but we're just interested in the total number of things which are on these plates. This is again for that 1 year period from October of 2011 to 1 year later. And you can see we started out with a very large number, which really confused me at that time. It then went down to relatively low levels, stayed low all the way through until the end of August, when we had a spike, and then it went back down again. That spike corresponded to Tropical Storm Isaac. Okay? We were very fortunate because we could sample here, which is this sampling date early in the cycle here, so this was relatively dry, normal rainfall. Then you can see Isaac came through, huge amount of rain happened, and we had a sampling date shortly after that. So we could see just the response of the river to the bacteria in the river to this rainfall event. And the questions then come, where is this coming from? If we look at the data splitting it out by northern sites and southern sites, for this one, this is the Isaac event. So these are the northern sites. And you can see there's far more bacteria here. So we probably have several things going on. We have a lot of water entering the canal system. Things are washing down into the Indian River. We also have, with these tremendous rains, a lot of turbulence in the river. So sediments are stirred up as well. So lots of things are happening. But the good thing is, one of the take-home messages here, is the river takes care of itself. So very quickly afterwards, we're back down to what are baseline normal levels of bacteria in the river. I'm interested in this stuff down the bottom as well. All these little low numbers. And of course, we can't see this on this scale because these are very high. So in order to see these levels, we have to change this to a logarithmic scale. So by changing that, we can start to see what's going on down at the bottom. This is also interesting if you just look at the red line, which is the southern sites, and the blue line, which is the northern sites. You can see the blue line is normally 5 to 10 times higher than is the red line. So the northern sites just carry a higher bacterial load than do the southern sites, where you have the better exchange. You have input from the canal system here. So what is going on? What bacteria are there? We don't know. But this is, again, projects we need to do in the future. So what are we doing? What are our future directions? We're continuing to do the monitoring. We want to know what's going on. We are developing these molecular source tracking techniques. We've got a project which we're just starting with Brian Lapointe. He was talking about this a few weeks ago, looking at the canal system in Vero Beach. So we're going to be getting his water samples, testing them to see what bacteria are present. You've also heard during this lecture series about the huge data sets which we have generated at Harbor Branch for many, many years, many sites. So we need to correlate our microbial data with these data sets. And we're also working to understand the types of bacteria and other microorganisms which are present in the Indian River Lagoon. Nobody knows what it is. It's one of the most biodiverse estuaries in the world, and yet we know nothing or practically nothing about the microbes. One of the projects we're working on in terms of microbial populations in the river involves pathogens. And for this, I'm going to turn it over to Gabby Barbarite, who's a PhD student in my lab, and she will talk for the next 10 minutes or so. 25 on Vibrios.
Oh, I can do 10. Here. Thank you. So where Peter talked about some of the bacteria that you want to have with you, I'm going to talk about the friends that you don't want to have with you in terms of bacteria. So before we get into that, I just wanted to start with what is waterborne disease. So we're switching gears here from the bacteria that are in the environment that humans are impacting to bacteria that are in the environment and how they're impacting humans. So before we get started, let's just define that. These are pathogenic microorganisms that are transmitted to humans through contact with water or some aquatic systems. And some of these things include bacteria, viruses, parasites, and they can be directed to us through things like drinking water, seafood, and activities like bathing and recreation. And the reason why I was roped into this talk is my research focuses on Vibrio Bacteria, and these guys are slightly curved gram-negative rods, and they're pretty diverse. The name Vibrio refers to their genus. It's a pretty large genus. It has about 30 species in it in total, and they have a pretty diverse range from bioluminescence all the way to disease. About half of the species that are in this genus are pathogenic, so they're able to cause infection. And in humans, these infections can be anywhere from seafood sickness, so just getting sick from eating something like a raw oyster, all the way up into pandemics like cholera. So very diverse. The three species that are implicated in the most disease in humans are vulnificus, parahaemolyticus, and cholerae. And even though these guys are pathogenic to humans, it's very important to understand that they are natural inhabitants of the marine environment. So we aren't putting them in there. They're there naturally. They just so happen to infect us. So they're not there as a result of pollution. And though they are natural, they exist in both virulent or infectious and non-virulent or non-infectious strains. And these guys aren't included in your normal standard monitoring for health departments. So in other words, if you want to look for them in a certain place, you have to go try to find them specifically. That's what our work is doing. So before we think of these guys as bad bacteria, I just want to say that they're in our environment for a reason. Their ecological niche, I can't do the nose crinkle thing that Peter does when he says it, but is actually to cycle nutrients. So they play a really big role in nutrient cycling, especially breaking down chitin, which is a compound that's produced by a lot of plankton. And it's estimated that over 100 billion pounds of chitin are produced in the oceans every year. So they do play a really important role in helping to cycle some of this. Now, of course, when they produce those toxins to break down chitin, when they do that to our cells, it doesn't turn out too good for us. But keep in mind that they do have a role in the environment. And these bacteria are constantly associated with some type of source. They have a really high substrate affinity. So we usually don't just see them floating around in the water. They're usually attached to something. So things like invertebrates that have these chitinous shells, as well as things like filter feeders that are constantly bringing in water. They say that oysters can shed over 100 million cells every day. So they can have pretty considerable impacts on the amount of bacteria in a certain area. These bacteria have also been associated with fish. So some studies in the Gulf have shown that habitat plays a really big factor on the type of bacterial load on these species. And for Vibrio particularly, we see that estuarine species like our redfish here has more of these bacteria associated with them than something like a mahi or a sailfish that lives offshore. We find these bacteria both internally and externally. Internally, it has a lot to do with their diet, so things that are feeding on these invertebrates that have these really high bacterial counts are going to have more in their digestive systems than things that are feeding on other fish. And we also find that their lifestyle has a lot to do with what's on the outside of their skin, so their dermal load. So a fish like this catfish that's rolling around in the muck and the mud is going to have a whole lot more of this bacteria on his skin than something like a barracuda that's swimming up in the water column. So the main thing that impacts where these bacteria are in the environment is salinity. So if we take a look at this graph along the bottom, we have salinity. 0 is freshwater, 30 is pretty much marine. And so looking at the top 3 species that infect humans, let's start with Vibrio parahaemolyticus. This guy tends to be at the top of the range. He tends to exist in more marine environments. Vulnificus here tends to be more brackish, middle of the road, and cholerae tends to be more at the fresh area, and that makes sense because cholerae is, of course, the causative agent of cholera, which is usually transmitted through drinking water. But we see that there's this really nice area of overlap right around the median salinities where all 3 species exist. So if you're looking for an area that could be potentially a good area to avoid, you would think that this would be a good area to start looking at. And so these things can be influenced both naturally by events like rainfall and drought, as well as anthropogenically with things like water release. So even though we're not putting these bacteria into the environment, we can do things to the environment that can select for these bacteria to appear or disappear. The other thing that influences their abundance is temperature. If we take a look here, we're looking at bacteria that are inside of an oyster. So we're looking at Vibrio parahaemolyticus in an oyster cell. We have the number of cells here along the top, and along the bottom we have locations. So we have the North Atlantic, Middle Atlantic, and the Gulf. And we can see very easily that these bacteria are much more abundant in the Gulf region, and that's directly as a result of the temperature. The waters here are very warm. These bacteria like warm water. We can also see another trend if we look at season. So we see winter, spring, summer, fall. All 3 of these have a peak right in the summer where these bacteria are the most abundant, again, related back to temperature. And these peaks in abundance in the environment, in an oyster in this particular example, also coincide with peaks in infections in humans. We usually see right around that time more and more people are becoming infected. So switching gears a little bit from the environment to humans, all human infections from these Vibrios are in some way traceable back to the marine environment. And we can be infected in a lot of different ways, which we'll go over in more detail in a little bit. But I just wanted to give you some stats off the bat here. These bacteria are responsible for about 80,000 illnesses in the United States alone every year, and they do occur all throughout the world. They're responsible for about 500 hospitalizations and ultimately about 100 deaths. The majority of these cases, about 67%, occur in the Atlantic and the Gulf regions. Again, this is going to be due to the warmer water as well as more people going out on the water. And Florida has the highest in the country, which isn't very surprising since it's pretty much summer here year-round, even though we do have 2 seasons. And we do have a lot of people that are, again, using this waterway system. So before we go any further, and I worry you guys too much, I just want to put the caveat out there that just because you're exposed to these bacteria doesn't mean that you're going to become infected. So the last thing I want you guys to do today is to leave and to not want to touch the water or to be afraid of going in the lagoon. Actually being infected depends on a lot of things. It depends on the type of exposure that you have, whether you ate some bad seafood or if you came in contact with it in the water. Depends on the dose of the bacteria that you encounter during this exposure, the virulence of them, whether or not they have the genes that can cause infection, and your individual health. Typical victims for these infections are usually males. There's something very interesting with estrogen that's going on that they haven't worked out quite yet, but the majority, 90% of victims are males. Usually they're over 60 years of age and are typically in some way immunocompromised. So if you're healthy, you probably don't have to worry about these things. Some of the predisposing conditions are things like liver disease, alcohol abuse, and diabetes. Of these general Vibrio cases that we see in the United States, more than half are gastroenteritis, which is basically just a stomach infection. This is usually from seafood. 24% are wound infection, and the remaining 17% are primary septicemia, which is a blood infection. And should you become infected, you need to treat it pretty quickly within the first 12 hours or so, and if you catch it early, you'll be prescribed some antibiotics. And depending on how far this progresses, some cases require surgical amputations. So the 2 most popular ways that people become infected by these are from eating raw seafood and through wound infections. So we'll start with seafood, since this is the majority of all of our U.S. cases. About 76% of all U.S. seafood sickness, all bacteria considered, are a result of Vibrio infections. And these bacteria are responsible for about 95% of all seafood-related deaths in the United States as well. Our culprit here is usually the oyster, the raw oyster. So about 15% of Americans consume oysters annually, but I'm not trying to cause too much alarm here because we only usually see an illness in every 10,000 meals. So if you like your oysters, that's fine, go at them. The culprit here is usually Vibrio parahaemolyticus. They're usually associated with these consumption cases. And so the average dose of parahaemolyticus that you take down in every yummy raw oyster is about 5.7 times 10 to the 4 cells per gram, and this is well within the range for infection. Of course, depends on the individual. And if you get your seafood from a good source, the FDA regulates that all commercially sold oysters have to sell stock that have less than 10 cells per gram, which is basically the lowest detectable unit for these bacteria. And they achieve this through post-harvest processing, either things like UV or temperature abuse. And of course, if you don't want to have to worry about any of these risks at all, just cook your oysters before you eat them. That'll get rid of all of these bacteria. So taking a look at some of the case reports of Vibrio parahaemolyticus here in the United States, the majority of the Vibrio infections here come from the Gulf of Mexico, which makes sense since about 70% of the US harvest comes from the Gulf. And again, this area has very warm water, and we usually see a peak in infections in the summer, again correlated with temperature. This area has been the focus of the majority of research for these bacteria just because it's so common there. However, for some of you guys that live up north, or even for people down here, this was quite a headline in the news over the summer, so you may have heard it, that they had what they were calling a parahaemolyticus outbreak up in the northeast coast off Massachusetts last summer. They had over 100 cases in 1 month, which was more than double what they normally have there during the summer. And it resulted in a lot of closures and recalls. And the media got a hold of this, of course, and they said that the reason why this was happening was global warming, and that these bacteria were moving into this area that they had never been before because the water was getting warmer. Well, I'm not going to touch that, but I will say that when I heard this, the first thing that I thought of was the necessity for a baseline, and that really inspired a lot of the work that we're doing here, because no one had actually looked for these bacteria in that area before in the environment. So when people got sick there, they said, oh, these bacteria showed up. But until we really look for them in the environment and describe where they are, we can't really say whether or not they were there. And so that's why it's really important to do these baseline projects, kind of like the one that we're about to get into here, so that when something like this happens, you can describe exactly what went on with what was going on in the environment. So the other way that people are infected is through injury, and this is usually a result of Vibrio vulnificus, which is, again, if you heard in the media, these are the flesh-eating bacteria that they were talking about last summer. These guys typically have a low infection frequency, only about 13% of all Vibrio cases. However, they're pretty severe. They have the highest hospitalization and fatality rates. So if you're going out in the water and this bacteria is there, there's really nothing to worry about. They've shown that the dermal load, or the amount of these bacteria that are on your skin if you're in the water, is only about 15% of what's out in the water. So the real problem comes down when you have a pre-existing injury, or basically a route for this infection to occur. So about 89% of the cases of people that have come down with this infection had some sort of pre-existing wound that sort of added a route for these bacteria to get into your blood. This bacteria has been nicknamed to cause fisherman's disease, and you can see why. Fishermen handle all sorts of things, sharp spines, sharp hook, teeth, bait, things like that, that can actually provide this infection. Looking at some of the cases of Vibrio vulnificus, they're very often related to fish. So the very first outbreak that they ever had was due to handling tilapia in a fish market in Israel. It was in the summer of 1999. 62 people were infected. There were no deaths. However, 70% required surgery, so it was pretty severe. Here in the United States, we haven't had any major outbreaks of that standard, but the only one that we've had was in Texas in July of 2004, and this was from a fishing competition. And there were 7 people that were infected, all from handling fish in the same area. Here in Florida, we haven't had any outbreaks per se. We usually have about 10 deaths a year in our state from this bacteria, Vibrio vulnificus. And some of you may have remembered that last summer there was quite a bit of press on this when a man that was fishing up in the Halifax River, just north of us in Daytona, became infected and died from this bacteria. And so the reason why he became infected was he, like some of the other cases that we talked about, had a pre-existing wound. I guess he had stepped in an ant pile or something like that. So he had that route there for these bacteria to get into his blood system. And what I took away from this when I heard this story was the idea of awareness. So his wife was quoted in saying that they had been here for their whole lives and been fishing here and never even knew that this threat or hazard was out there. And so that kind of inspired some of our work too, to try to tell the public about these risks when they're going in these areas. And that brings us back here to the Indian River Lagoon. So as you know, this area covers about 40% of the Florida East Coast. It's a very significant estuary, though if you came to Grant Gilmore's talk last week, you know that we don't know very much about the microbial communities in our area. However, it makes sense to look for these bacteria here, as we have very similar conditions to places like the Gulf, temperature and salinity, as well as species, so some of the same things like the oysters and the fish. And we also have anthropogenic influences and a very dense coastal population. And this coastal population is definitely using this area to the fullest. It's a fishing hotspot. Over 50% of the mainland trips occur in the IRL, and it generates quite a bit of revenue for us locally. So if we look here at the Vibrio cases that have been reported from our region, looking at this map here, we have the 6 counties that border the lagoon with Volusia at the top and Palm Beach at the bottom. This whole area collectively is responsible for about 20% of all Vibrio infections within Florida. The majority of our infections are usually in the Gulf, but since we're focusing in on the lagoon here, we've had an increase in the number of cases here in our region. And as we click through the map, I want you guys to focus that the darker the area, the higher the rate of infection for that area is. So we're going to click through here, and you see that as we go from 1995 to 2005, 2005 to 2012, this area gets a lot darker. So we're seeing that infection frequency is increased in our area. Now, this could be due to a lot of things. It could be due to things like better reporting. So now we have better surveillance. People are more aware of this. Of course, after that story made the news last summer, I'm sure everyone that had a bug bite or a stomach infection was going to their doctor and asking if they were okay. This can also be an effect of more people going in the environment, into the water. So more and more people are coming to visit warm, sunny Florida and are going fishing in the lagoon. But from this, we can conclude that these bacteria are having some potential impact on local communities. And that was sort of the jumping off point that we came to for starting up this research project. And so my project specifically, our first objective is to document the occurrence of these bacteria in the Indian River Lagoon. And we want to be able to answer for you where and when can you become infected by these. Our second objective is to look for these bacteria with commonly contacted sources. And from this, we want to be able to tell you how can you become infected. And our last objective here is to evaluate the virulence or the potential for them to infect you. And for this, we're only really looking at vulnificus and parahaemolyticus. We're not as focused on Vibrio cholerae just because we don't usually have pandemics like cholera here. And our goal with this is to tell you what your potential or your chances are of becoming infected from these bacteria. So where are we looking in the lagoon? Well, let's start with Harbor Branch here. So you are here. This is a really great area for us, not only because it's convenient, but also because this sort of serves as a reference site. It's generally non-impacted. There's not a lot of development around here. So you would expect that it would be pretty clean water. But if you remember, that area of overlap with those 3 species was all in that brackish range. So we've really honed in on these spillway sites. So areas where we have constant fresh water and lagoon water mixing to create that perfect brackish salinity. And so the majority of our sites are going to be focused at areas like that, since that's where we would expect to find all 3 species. And we've included a final site that's slightly outside of the lagoon that's going to be fully marine environment, and that's here right outside the Fort Pierce Inlet that we would expect to be outside their range. So how are we finding these bacteria? Well, we're trying to work out some of the hot spots or where they're found and when they're found by going out every month to all of these sites and collecting sediment. Now, sediment is a great indicator for these things because sediment doesn't move. Water moves, fish move, sediment stays in one place. So we can take that back to the lab and get a really good idea of what's happening locally in that area. Then we go back to these sites and we're collecting things that people normally come into contact with. So things like fish, and we do that by taking a little clip from their fin, and we can grow up the bacteria from there. Other things that you could potentially be handling or getting hurt by, a hook or bait, things like sharp crab claws and little rostrums on shrimp. And then things like oysters. People are shucking oyster shells and eating oysters, so we're sampling those too. Back in the lab, we bring our sample in, and we enrich it overnight to grow these guys up, and then we plate it out, just like Peter showed you. And we're using a very good selective media, and the reason why this one's so helpful is that each of our 3 target species grow up a different color. So I can look at my plate pretty easily and say purple is parahaemolyticus, teal is vulnificus, and blue is cholerae. So I can look at my plate and get a really good idea of what's going on at that spot. The second wave of this is that we can extract DNA from these strains, and then we can actually do a genetic test, a molecular test known as PCR, polymerase chain reaction, where we can look at whether or not that particular strain has the genes to cause infection in humans. So we have it for vulnificus and parahaemolyticus. We see an environmental strain here that doesn't have genes to infect you, and a clinical strain that does. So not only can we see how much of this bacteria is in the environment, but whether or not it's a potential hazard. So what have we found so far? Well, first off, we're the first ones to ever look for this bacteria here. So that's kind of cool when you think of all the other biodiversity studies that have been done here and that no one's actually said, yes, this bacteria is here. We know people get infected, but no one's ever found it. So we found, of course, what we were expecting, that at these pink spillway sites, we found all 3 species. At our Harbor Branch site, however, we only found parahaemolyticus, but that kind of does make sense because they're typically at the higher salinity range, and so that this area doesn't have a spillway, so that made sense. And what I thought was really cool is that we didn't find any offshore in the Fort Pierce Inlet, and I kind of smiled to myself when all this stuff came out in the press and they were saying, don't go in the ocean because you'll get infected by this flesh-eating bacteria. No, you won't. So, which is another good reason why the press and the scientists should communicate. But, so yes, we were able to say that these bacteria were present in the Indian River Lagoon. There was some variation by location. Are there potential hotspots or areas that you can go where your chances of becoming infected are higher? Well, stay tuned. Hopefully in the next year or so, we're going to work that out. The next wave of this was to look for the different sources and how they carry these bacteria. So we found it in the sediment, so that gave us the green light to say, okay, it's here in the lagoon. Now let's see who else is carrying it. We found all 3 species in oyster. And then the fish is where it gets a little bit interesting. And I've been a fish nerd, so I like this stuff. But our catfish had all 3 species. Our mullet only had parahaemolyticus associated with his skin, as indicated by those fin clips that we were taking. And our barracuda and jack so far haven't had any. And that's kind of interesting because the catfish and the mullet are rolling around in the muck, where our barracuda and our jack are up in the water column. So you do see variation between different species and what they're carrying on their skin. We also found these pathogens on bait shrimp, which is interesting because people are using them to fish. So we found them associated with multiple sources, and we saw some variation. And again, this is very preliminary data, but we're going to work out some of these details in the future, hopefully get a bigger subset of fish. And so the last wave of this is to look at the virulence. And the virulence, again, is the ability of it causing infection in human. And this is a genotype that we're looking for that basically has the ability to produce toxins and lyse human cells, so not other animals. We're looking specifically for humans. And so before you look at this and see tons of red and freak out, just say that there's always a mix of both strains in the environment. So even normally when people aren't becoming infected, you see mixes of clinical and environmental strains. That's just normal. Remember, these are some of the things that are helping them break down those chitin cells that they do normally. So if we take a closer look here at Riverside Park in Vero, some of the things that we collected from that site, again, we see that there's this mix of clinical and environmental strains. And again, there's no reason to really be alarmed by it because in some of the other literature we find that, well, this was taken in August, and that's usually when people are infected, so these bacteria are very abundant in the environment. They're breaking down chitin and cycling nutrients and things like that, and that's when we got this data that we got here. However, it could be a potential hazard for people that are using the lagoon, and so while we don't have all the answers on that yet, we do plan to continue that, investigating that more in the future. So what have we found so far? Again, our study was the first to document these bacteria in the Indian River Lagoon. We're going to continue evaluating some of the different places and the different times of year, as well as the sources and how these strains can potentially infect you. And we hope to share our findings with things like managing agencies and medical care providers and IRL users. So our biggest goal here is to make your time on the water as safe and enjoyable as possible. And the part of the talk that I'm sure you guys are all interested the most in is how can you actually avoid these infections? Well, for the wound infections, be aware of your local hazards and minimize your exposure. So if you hear in the news that these guys are abundant in a certain area, simply don't go there. Avoid injuries by using protective gear. So if you're shucking an oyster, wear a glove. If you do happen to get a puncture wound from, slipping on a piece of glass or poking yourself with a fish hook, just wash it out. Chances are you'll be fine. And as far as seafood goes, avoid eating raw oysters if you have any of those predisposing conditions. So talk to your doctor if you have some of those, liver disease, and ask them if it's safe for you to eat oysters. Minimize your raw oyster consumption during warm months when these bacteria are most abundant in the environment. It's just a good idea generally. And always purchase your seafood from a reputable source so that you know it went through the appropriate post-harvest processing and don't have that bacteria associated with them. Last, if you think that you're infected, seek care quickly. And when in doubt, treat it like it's infected, whether it's a fish or a wound or something that you're about to eat. If you treat it like it's infected, chances are, again, that you'll be fine. And so with that, we'd like to thank our funding sources as well as some of the other people. You want to do the honors?
No, you can. You're doing so well.
Oh, so well. Well, thank our lab, Kath and Dee. Kath's in the back. She was in that video. As well as the Marine Mammal Department. Adam Schaefer does some work in our lab. Some of the interns that have helped with this work, Amber, Lexi, and Carly, as well as our volunteers, Martha and Luke, and David, who collected all of my fish for me and was roped into this project.
He had to. He had no choice.
David was married into my project. As well as Dennis's group who collected some of our samples for us, and Dr. Anita Wright, who is my contact up at the University of Florida, who's been doing a lot of this work in the Gulf and kind of gave us some of our methods. And the license plates?
License plates and Stuart,
who funded
supported
some of our
this work. So thank you for your attention.
And with that, we'll take